So student, welcome. In last lecture, we have learned about the structure of sperm, structure of ovum. Then we have seen the fertilization process. Means we have learned about the zygote stage. So in female genital tract, in fallopian tube, in the ampular region, there is the union or there is the fusion of male gamete and female gamete, and there is formation of zygote. There is formation of zygote. This zygote is diploid one. This zygote is diploid one. As soon as zygote is formed, there is division and redivision, and mass of cell is formed. There is division and redivision, and mass of cell is formed, and there is formation of germinal layers. But all this process goes through 4 steps. These 4 steps are, first of all, it is cleavage. Then second stage is blastulation. Third stage is implantation. And fourth stage is gastrulation. Means when zygote is formed, up to 4 days, it is in the fallopian tube. After 4 days, it enters the uterus, and in uterus, up to 7th day, there is the implantation. At near about 7 days requires for the implantation, and before implantation there is blastulation of the zygote. There is a blastulation, and after blastulation there is a blastocele form, blastomeres form, and then cavity is formed. Then trophoblast cells are formed. Zona pellucida is there, and then after that process, zona pellucida gets dissolved, and there is an implantation process with the help of trophoblast inside the trophoblast. It penetrates, or it forms a burrow in the uterine wall or in the endometrium, and this foetus or this blastocyst get implanted in the endometrium, and after the implantation there is next step, that is gastrulation, and at the end of the gastrulation 3 germinal layers are formed. So we will see now step by step. First of all, cleavage process. First of all, we will see cleavage process. Now, when zygote is formed, this is the zygote, which is diploid one. This is a zona pellucida covering. Zona pellucida. This is formed in ampular region, ampulla of fallopian tube. Then after this, what happens? The next process starts. And there is a horizontal division. And 2 cells are formed. Here 2 cells are formed. There are 2 polar bodies. These polar bodies can be seen here. These are polar bodies. So inner mass of cell or inner cell protoplasm divides into 2 cells. These are 2 cells. These are polar body. This is zona pellucida here. Then after that, there is a next division. This is a 2 cell stage. This is the 2-cell stage. Then after this, again there is a division. Again there is a division at the right angle of first division. At the right angle of first division, and 4 cells are formed. This is the 4-cell stage. This is the 4-cell stage. 4-cell stage. Then from this 4-cell stage, again there is a division and 8 cells. Now these cells in this particular zygote are called as blastomeres. Now we will not call it zygote, we will call it now embryo. This is embryo. The next development starts like this. Then from this 4-cell stage, again this cells divides. Again there is a division, and from 4 cells, 8 cells are formed. From 4 cells, 8 cells are formed. So this is the 8-cell stage. Then from this, again there is a division, where there is a repeated division, and from this 8-cell stage, we can see here near about 16 cells are formed. Double these cells. 16 cells are formed. Then again there is a division. Again there is a division, and near about 32 cells are formed. And these 32 cells, if you see in it, outer cells, we can see them somewhat smaller cells, and inner somewhat larger cells. Smaller cells outside, they are called as clearer cells, and inner are larger cells. So near about 32 cells are formed. Near about 32 cell stage, and this 32 cell stage is called as morula. This 32 cell stage is called as morula. Why it is called as morula? Because it looks like the fruit of mulberry. It looks like the fruit of mulberry, and so this is morula stage. And these are the larger, smaller, clearer cells. Smaller, clearer cells. And these are the inner, larger cells. Outer, smaller cells. And the cells included in all, they are called as what? Blastomeres. They are called as what? Blastomeres. This is zona pellucida. One point you should note here, that the size of embryo does not changes. All these stages are completed near about in 4 days. In 4 days, the cell of the size of the embryo does not changes. Only what changes? The size of the cell. Only there is a change in the size of the cell. They get smaller and smaller as there is a repeated division. As there is a repeated division, the cells become smaller and smaller. So, in this stage, in this particular stage, we have seen that in cleavage stage, we have seen that from zygote, 2 cell stage is formed. Then there is right angle division to the first division, 4 cell stage is formed. Then from 4 cell stage, again there is a division horizontally and 8 cell stage is formed. Then there is again division, 16 cell stage is formed. From there is again division, 32 cell stage is formed. And in the 32 cell stage, the cells are arranged in 2 layers. Outer one are smaller in one and inner one are larger cells. So at this stage, all these cells included in this stage is called as blastomeres, and this 32 cell stage is called as what? Morula. Why it is called as morula? It looks like the fruit of the mulberry, so it is called as morula. So this is the cleavage. This is the cleavage. Now we will see the second type, blastulation. Second step, blastulation. Blastulation. When there is a formation of blastocyst from the morula, then that is called as what? Blastulation. That is called as what? Blastulation. What happens? The 32 cell stage of morula. 32 cell stage of morula. What happens in it? The size of the cells increases. The size of the cells increases. They are arranged in 2 layers. Now this outer smaller cells, a border-like cells is formed, and this is called as trophoblast. This is called as trophoblast. Then the blastomeres are arranged at one end. They get gathered at one end. They get gathered at one end. These are called as trophoblasts. This is still attached zona pellucida. These are called as embryonal cell. Inner one which are attached to the trophoblast cells. They are called as embryonal cell. And this end is also called as embryonal end, or it is called as animal pole. And opposite to this pole is called as vegetal pole. This pole is called as vegetal pole. This is called as animal pole. Where embryonal proper cells are present, that pole is called as animal pole, and opposite to it is called as vegetal pole. Then, has the size of these cells begin? This, because after 4 days, this morula enters the uterus, and after entering in the uterus, it derives the uterine milk, which is rich in glycogen. So the fluid enters it enters in the morula and it pushes the cell size also get double. So there is a pressure on zonopellucida, and zonopellucida layers get destroyed or they get ruptured, they get out degenerate, and we can see that from this structure naked trophoblast cells we can see. Zonopellucida get degenerated because of the pressure of blastocele, because uterine milk enters into blastocyst, and the blastocyst the trophoblast cells comes out from the zona pellucida. These are the trophoblast cells. Now this structure has lost cells of Rauber. Embryonal cell, or it is also called as embryonal proper, embryonal cell. And the cells opposite to these are called as hypoblast cell, embryonal cell. So there is no need of learning this. We have not that structure in our textbook. So trophoblast cells, these are the trophoblast cells. This is the outer layer. So due to the uterine milk, and this cavity is formed, that is called as what? Blastocele. Why this cavity is formed? Because this blastocyst, we can call this as blastocyst. This blastocyst absorb the uterine milk which is present in the uterus, and due to the pressure of it, the cell size get doubles. This cell size get doubles, so there is a pressure inside, and a cavity is formed. And that cavity is called as blastocoel, and the cells inside this trophoblast get arranged to the one end. And the point at which this inner mass of cells get attached to trophoblast cells, that trophoblast cells are called as cells of Rauber, and that end is called as at which embryonal cells are attached, that end is called as embryonal end or animal pole, and opposite to embryonal pole, that pole is called as what? Abembryonal end. Abembryonal end, or that pole is called as what? Vegetal end. That pole is called as vegetal end. So this is blastocyst. So blastolation process ends up to the 7 days. Means 1 to 4 days the embryo is present in fallopian tube. After 4 days, it enters the uterus. Up to 7 days, this embryo is suspended in uterus. It does not get implanted. So, this suspended embryo sucks the blood, gets the uterine milk, the glycogen which is present in the uterus, it nourishes it, and the size of the morula get double, and due to this pressure a blastocele is formed. The cells, mass of cells get transferred to one end. That end is called as embryonal end, and it get attached to the trophoblast cells. That trophoblast cells that point of trophoblast cell at which embryonal cells get attached, that is called as cells of Proudhon. And that embryonal cells opposite to embryonal cell, embryonal end, that end is called as what? Abembryonal end. So such is the blastulation stage, and it is completed near about 7 days, up to 7 days after fertilization. After fertilization, now this blastocyst is ready for implantation after 7 days. This blastocyst is ready for implantation after 7 days. Now we will see the next stage, that is implantation. That is implantation. Now what is implantation? We know. We see that when we take the small plant and plant in the field, then we call it as a plantation. Here implantation, when embryo is set, embryo is placed in a uterine cavity or in the uterus, in the wall of uterus, and that wall of uterus is called as what? Endometrium. That wall of uterus is called as what? Endometrium. We have seen the layers of the uterus, that is endometrium, myometrium and perimetrium. Endometrium is the innermost layer. It is the innermost layer. So when there is a plantation of embryo in the endometrium, then that is called as what? Implantation. That is called as what? Implantation. There is the blastocyst, gets attached to endometrium of uterus, and it is attached to fundus point. Means because we see that uterus has 3 regions. The dome shaped structure is called as fundus. This is the dome shaped structure. This is called as fundus. This is called as body, and this is called as cervix. So at this fundus point of uterus, this blastocyst get attached after 7 days. Near about 7 days, it begins to implant it. And what happens? The cells of trophoblast, these cells of trophoblast forms 2 layers. Trophoblast cells forms 2 layers. One syncytio, trophoblast, and second one is cytotrophoblast. These syncytotrophoblast cells, what happens? They starts to enter, they form a structure which penetrates or forms bureau in endometrium, form bureau or penetrates endometrium. They are sticky and active. They have an ability to form. So if this is a trophoblast, such structures are formed which can enter or which can penetrate in the endometrium. So syncytiotropoblast, with the help of syncytiotropoblast, this blastocyst get implanted in the fundus region of uterus or in the endometrium of uterus, and it get implant due to the activity of syncytiotropoblast. Cytoplasm is the inner layer of trophoblast. The size of the cell does not increase. It is retained at that position. Only what happens, syncytiotropoblast forms a structure which penetrates into the endometrium, and with this structure, this structure trophoblast cells, what they happen, they derive the uterine milk from the endometrium. And completely, these blastocysts completely get buried in the endometrium. So this is called, when these blastocysts get completely buried in the endometrium, then we can say that these blastocysts is completely implanted in the uterine cavity or uterus. So this is called as implantation. The next point we will see tomorrow. Thank you.